Thin layer chromatography. Thin layer chromatography is a chromatography technique that separates components in non-volatile mixtures. It is performed on a TLC plate made up of a non-reactive solid coated with a thin layer of adsorbent material. This is called the stationary phase. The sample is deposited on the plate, which is eluted with a solvent or solvent mixture known as the mobile phase. This solvent then moves up the plate via capillary action. As with all chromatography, some compounds are more attracted to the mobile phase, while others are more attracted to the stationary phase. Therefore, different compounds move up the TLC plate at different speeds and become separated. To visualize colorless compounds, the plate is viewed under UV light or is stained. Testing different stationary and mobile phases is often necessary to obtain well-defined and separated spots. TLC is quick, simple, and gives high sensitivity for a relatively low cost. It can monitor reaction progress, identify compounds in a mixture, determine purity, or purify small amounts of compound. Procedure. The process for TLC is similar to paper chromatography but provides faster runs, better separations, and the choice between different stationary phases. Plates can be labeled before or after the chromatography process with a pencil or other implement that will not interfere with the process. There are 4 main stages to running a thin layer chromatography plate. Plate preparation. Using a capillary tube, a small amount of a concentrated solution of the sample is deposited near the bottom edge of a TLC plate. The solvent is allowed to completely evaporate before the next step. A vacuum chamber may be necessary for non-volatile solvents. To make sure there is sufficient compound to obtain a visible result, the spotting procedure can be repeated. Depending on the application, multiple different samples may be placed in a row the same distance from the bottom edge. Each sample will move up the plate in its own lane. Development chamber preparation. The development solvent or solvent mixture is placed into a transparent container to a depth of less than 1 cm. A strip of filter paper is also placed along the container wall. This filter paper should touch the solvent and almost reach the top of the container. The container is covered with a lid and the solvent vapors are allowed to saturate the atmosphere of the container. Failure to do so results in poor separation and non-reproducible results. Development. The TLC plate is placed in the container such that the sample spot are not submerged into the mobile phase. The container is covered to prevent solvent evaporation. The solvent migrates up the plate by capillary action, meets the sample mixture, and carries it up the plate. The plate is removed from the container before the solvent reaches the top of the plate, otherwise, the results will be misleading. The solvent front, the highest mark the solvent has traveled along the plate, is marked. Visualization. The solvent evaporates from the plate. Visualization methods include UV light, staining, and many more. Separation process and principle. The separation of compounds is due to the differences in their attraction to the stationary phase and because of differences in solubility in the solvent. As a result, the compounds in the mobile phase compete for binding sites on the stationary phase. Different compounds in the sample mixture travel at different rates due to the differences in their partition coefficients. Different solvents, or different solvent mixtures, gives different separation. The retardation factor, or retention factor, quantifies the results. It is the distance traveled by a given substance divided by the distance traveled by the mobile phase. In normal phase TLC, the stationary phase is polar. Silica gel is very common in normal phase TLC. More polar compounds in a sample mixture interact more strongly with the polar stationary phase. As a result, more polar compounds move less while less polar compounds move higher up the plate. A more polar mobile phase also binds more strongly to the plate, competing more with the compound for binding sites. A more polar mobile phase also dissolves polar compounds more. As such, all compounds on the TLC plate move higher up the plate in polar solvent mixtures. Strong solvents move compounds higher up the plate, whereas weak solvents move them less. If the stationary phase is nonpolar, like C18 functionalized silica plates, it is called reverse phase TLC. In this case, non-polar compounds move less and polar compounds move more. The solvent mixture will also be much more polar than in normal phase TLC. Solvent choice. An allotropic series, which orders solvents by how much they move compounds, can help in selecting a mobile phase. Solvents are also divided into solvent selectivity groups. Using solvents with different elution strengths or different selectivity groups can often give very different results. While single solvent mobile phases can sometimes give good separation, some cases may require solvent mixtures. In normal phase TLC, the most common solvent mixtures include ethyl acetate, hexanes for less polar compounds, and methanol, dichloromethane for more polar compounds. Different solvent mixtures and solvent ratios can help give better separation. In reverse phase TLC, solvent mixtures are typically water with a less polar solvent. Typical choices are water with tetrahydrofuran, acetonitrile, or methanol. Analysis. As the chemicals being separated may be colorless, several methods exist to visualize the spots. Placing the plate under black light makes fluorescent compounds glow. TLC plates containing a small amount of fluorescent compound in the adsorbent layer allow for visualization of some compounds under UVC light. The adsorbent layer will fluoresce light green, while spots containing compounds that absorb UVC light will not. Placing the plate in a container filled with iodine vapors temporarily stains the spots. They typically become a yellow or brown color. The TLC plate can either be dipped in or sprayed with a stain and sometimes heated depending on the stain used. Many stains exist for a large range of chemical moieties, but some examples include potassium permanganate, ninhydrin, acidic vanillin, phospholipdic acid. In the case of lipids, the chromatogram may be transferred to a polyvinylidene fluoride membrane and then subjected to further analysis, for example, mass spectrometry. This technique is known as Far Eastern blot. Plate production. TLC plates are usually commercially available, with standard particle size ranges to improve reproducibility. They are prepared by mixing the adsorbent, such as silica gel, with a small amount of inert binder like calcium sulfate and water. This mixture is spread as a thick slurry on an unreactive carrier sheet, usually glass, thick aluminum foil, or plastic. The resultant plate is dried and activated by heating in an oven for 30 minutes at 110 degrees Celsius. The thickness of the absorbent layer is typically around 0.1 to 0.25 mm for analytical purposes and around 0.5 to 2.0 mm for preparative TLC. Other adsorbent coatings include aluminium oxide or cellulose. Applications. Reaction monitoring and characterization. TLC is a useful tool for reaction monitoring. For this, the plate normally contains a spot of starting material, a spot from the reaction mixture, and a co-spot containing both. The analysis will show if the starting material disappeared and if any new products appeared. This provides a quick and easy way to estimate how far a reaction has proceeded. In one study, TLC has been applied in the screening of organic reactions. The researchers react an alcohol and a catalyst directly in the co-spot of a TLC plate before developing it. This provides quick and easy small-scale testing of different reagents. Compound characterization with TLC is also possible and is similar to reaction monitoring. However, rather than spotting with starting material and reaction mixture, it is with an unknown and a known compound. They may be the same compound if both spots have the same RF and look the same under the chosen visualization method. However, co-elution complicates both reaction monitoring and characterization. This is because different compounds will move to the same spot on the plate. In such cases, different solvent mixtures may provide better separation. Purity and purification. TLC helps show the purity of a sample. A pure sample should only contain one spot by TLC. TLC is also useful for small-scale purification. Because the separated compounds will be on different areas of the plate, a scientist can scrape off the stationary phase particles containing the desired compound and dissolve them into an appropriate solvent. Once all the compound dissolves in the solvent, they filter out the silica particles, then evaporate the solvent to isolate the product. Big preparative TLC plates with thick silica gel coatings can separate more than 100 mg of material. For larger scale purification and isolation, TLC is useful to quickly test solvent mixtures before running flash column chromatography on a large batch of impure material. A compound elutes from a column when the amount of solvent collected is equal to 1 RF. The eluent from flash column chromatography gets collected across several containers called fractions. TLC helps show which fractions contain impurities and which contain pure compound. Furthermore, two-dimensional TLC can help check if a compound is stable on a particular stationary phase. This test requires 2 runs on a square-shaped TLC plate. The plate is rotated by 90 before the second run. If the target compound appears on the diagonal of the square, it is stable on the chosen stationary phase. Otherwise, it is decomposing on the plate. If this is the case, an alternative stationary phase may prevent this decomposition. TLC is also an analytical method for the direct separation of enantiomers and the control of enantiomeric purity, e.g. active pharmaceutical ingredients that are chiral.